Welcome to the Bio Whisperer channel, and the topic today is on a perspective overview of CRISPR antimicrobial resistance targeting the escape. Since the success rate for the development and approval for new antibiotics is very low, where a new class of antibiotics has not been discovered for several decades. There was a time when all of our antibiotics came from natural sources. For instance, chloramphenicol, made by a bacterium found in compost in Venezuela, and then chlorotetracycline, excreted by a bacterium in soil samples. After a long discovery drought in soil sample analyses, drug firms stopped looking for new antibiotics in soil samples, and focused on creating synthetic compounds in laboratories. The premise remains that the challenge is to find things that kill bacteria but don't harm the person taking them. Some of the approaches include using bacteriophages that target and lyse the targeted pathogenic strains. Notably, artificially derived synthetic peptides or enzymes have been quite successful against some of the resistant strains. Given the widespread use and abuse of antimicrobial agents, resistance due to continued genetic changes is likely to be a concern tomorrow. Based on our understanding thus far, a small number of the pathogens are responsible for most antibiotic-resistant infections, known by the abbreviations ESCAPE. The 6 key members are as follows, starting with Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and lastly Enterobacter. These bacteria have diverse post-translational modifications that are involved in the pathogenesis. PTMs for the escape pathogens are essential to identify the host cell pathways that are targeted, and this strategizes a possible win against these bacteria by controlling the spread of their infectivity and virulence. Bacteriophages are bacteria-specific viruses, which can specifically infect and lyse bacteria. Phage therapy harnesses phages for treatment against bacterial pathogens and their infectious disease. Suggestions on deploying metabolic inhibitor properties of phage structural proteins to inhibit bacterial cell wall synthesis, as well as the use of bacteriophages as a carrier and vehicles for the delivery of engineered genomic materials and vaccine antigens. Alternative research strategies such as the development of nucleic acid-based antibacterial treatments, antibacterial peptides, bacteriocins, antivirulence compounds, and bacteriophage therapies should be exploited to cope infections caused by resistant superbugs. Antimicrobials are life-saving medications, but only if they work against the organism causing infection. AMR may impact on life-saving health care, such as cancer treatments or organ transplants, if antimicrobials are not able to be effective in preventing infections that are commonly associated with these procedures. The World Antimicrobial Awareness Week is celebrated from 18th to 24th of November every year. The 2021 theme is Spread Awareness, Stop Resistance. Antimicrobial resistance is already happening and unfolding as a worldwide problem that affects human and animal health. The campaign has been successful in raising awareness regarding the misuse of antimicrobials contributes to antimicrobial resistance, and in particular scenarios whenever antimicrobials must be used, they must be used with care. CRISPR-Cas system to promote sensitizing bacteria to antibiotics. CRISPR-Cas is an RNA-guided DNA cutter where the role is to cleave foreign genetic material upon bacteriophage infection inside the bacteria. Given its sequence-specific targeting, which refers to the ability to distinguish between commensal and pathogenic bacterial species. Guide CRISPR RNA can be constructed to target only chromosomal and virulence genes that are highly specific to pathogens. CRISPR guide RNA can be designed specifically to target resistance or virulence genes. It will induce a break inside the dsDNA of resistant bacteria, reverting them into the antibiotic-sensitive ones, a process known as sensitizing bacteria to antibiotics. Development of smart antibiotics that can differentiate between beneficial and pathogenic microorganisms, in turn giving hopes towards eliminating multi-drug-resistant pathogens. Henceforth creating opportunities in the treatment of multi-drug-resistant infections. CRISPR-Cas systems have been identified as a bacterial adaptive immune system. The many uses includes genome engineering, genome editing for treating medical disease, reversal of antibiotic resistance by targeting resistance genes. Using a pair of GR-directed Ca nucleases instead, it is also possible to induce large deletions or genomic rearrangements, such as inversions or translocations, which can site-specifically cleave double-stranded DNA, resulting in the activation of the double-strand break repair machinery. Double-strand breaks can be repaired by the cellular non-homologous end joining pathway, resulting in insertions and/or deletions, indels, which disrupt the targeted locus. In this context, CRISPR-Cas has been used to target specific genes in virulence and genes that encode antibiotic resistance in bacterial populations. CRISPR-Cas system has been viewed to have a role towards neutralizing antibiotic-resistant genes where CRISPR-Cas-based nucleases as sequence-specific antimicrobial agents could reduce the relative abundance of the target and infectious bacteria, yet the development of a broadly applicable and robust delivery system remains a key milestone. Currently, most of the research effort is focused on the phage, conjugative, and polymeric nanoparticles-based CRISPR delivery systems. As a new weapon, within the DW article, it is mentioned that scientists used CRISPR-Cas to target an antibiotic-resistant gene, and modified it to be much more transferable between bacteria. Subsequently, they put it inside harmless bacteria and fed them to the mice. Surprisingly, it eliminated more than 99.9% of the targeted antibiotic-resistant bacteria after only 4 days. Though CRISPR-Cas is a powerful and precise tool with the potential to be very helpful in eliminating antibiotic resistance, researchers don't yet know if bacteria are also capable of developing resistance to this technology. Given that hotspots of antibiotic resistance gene transfer, such as wastewater treatment plants or livestock farms, will always be there, where the potential to reduce transfer of resistance genes into human pathogens. Based on what we have illustrated thus far, a process called horizontal gene transfer, it is possible that antibiotic resistance genes from harmless environmental bacteria can find their way into more dangerous human pathogens by hitchhiking on genetic elements such as plasmid. The premise of CRISPR's role in antimicrobial resistance is to firstly program Cas to target an antibiotic resistance gene found on a plasmid. The guide RNA enzyme system will cleave the DNA at its appropriate sequence. This stops plasmid replication and leads to removal of the plasmid and its AMR gene from the bacterial cell and its progeny, resensitizing the bacteria to antibiotics. In short, removing by deactivating the antibiotic resistance gene is achieved. Taken broadly, we see an equally important role for new antibiotics discovery. We know it's really difficult and needs some luck. More importantly, what we can do at the present moment is responsible stewardship, which includes the unnecessary use and preventing abuse of antibiotics in scenarios where it is not required. However, not all is lost, we do see some light at the end of the tunnel. Recently, in November 2021, new research published describes how CRISPR-Cas gene editing can be used to create new non-ribosomal peptide synthetase enzymes that deliver clinically important antibiotics. Non-ribosomal peptide synthetase enzymes are prolific producers of natural antibiotics such as penicillin. However, up until now, manipulating these complex enzymes to produce new and more effective antibiotics has been a major challenge. It is envisaged that this approach will lead to new assembly lines that can deliver new peptide products. We should be able to anticipate more research in this hotspot in the near future. In another research report in November 2021, the team from UCSF reported success in modifying bacteria already thriving within the digestive system. Using the gene editing tool to target and kill one strain of E. coli while leaving another alone, the researchers managed to achieve it by using a virus, called M13, to inject a CRISPR-Cas9 system into the cells of a specific strain of E. coli, where it went to work chopping out segments of DNA. It is mentioned within the report that the research findings stands to advance our understanding of the microbiome and could eventually pave the way for treating gut-related disease. Thank you for watching this video. If you like this video, please smash the like button and subscribe to this channel for future updates.